Okay, so I'm going to pick up where we left off in chapters 25 and 26, talking about the eukaryotes. Specifically, in this sort of group of lectures, we're going to be talking about what they are, what traits contribute to their success, when they first appeared in the geological history of Earth, and how did they evolve? How did they arise? So remember, we're talking about this domain here, the eukaryotes. So we had this list of unique traits that the eukaryotes have that contribute to their success. We talked about larger cell size, and really this idea that eukaryotic cells in general tend to be much larger than prokaryotic cells, like the one you see in the drawing here. And the larger size of eukaryotic cells made way for room for things like a cytoskeleton and organelles, a nucleus, and all of these things made way for things like sexual reproduction and multicellularity. So I haven't gotten into the details of what all of these numbers 2 through 6 items are, but know that larger cell size helped make way for these other important traits unique to the eukaryotes to evolve. And let's talk about why each of these individual traits here is important, because one thing we talked about is that larger cell size is cells or organisms get larger and larger and larger, their surface area to volume ratio gets lower and lower and lower. And that lower surface area to volume ratio means that simple diffusion is not enough to get all the wastes into or out of all the regions of a eukaryotic cell and all the nutrients into all the regions of a eukaryotic cell, because there's just not enough surface area relative to the volume for simple diffusion to move those things around to all parts of the cell. And so it's important to point off that this larger size is what's known as a trade-off. So a trade-off in the sense that having that larger size allows all those other adaptations, things like organelles, and we'll talk about why those things are important, to fit into the cell. But it's a trade-off in the sense that all of those things also, it's sort of a chicken and the egg problem in the sense that all those things needed to evolve to stop a eukaryotic cell from starving or waste building up too much in a eukaryotic cell. So let's talk about some of the ways eukaryotic cells deal with this sort of trade-off and this issue of this lower surface area to volume ratio. And the first thing I want to talk about is something called a cytoskeleton and the system of membranes that are inside eukaryotic cells. And I'm generally going to refer to this as just the overall internal organization, and the fact that there is a lot of unique adaptations in internal organization in eukaryotic cells. And when I talk about all of these unique characteristics, things like organelles, a cytoskeleton, a nucleus, sexual reproduction, multicellularity, these are all unique game-changing innovations that eukaryotic cells came up with. So you can kind of think of eukaryotic cells as the Bill Gates of the cellular world, where they come up with these really unique game-changing innovations that really change the whole way that organisms work. So let's talk about that first unique major innovation that eukaryotes have evolved in terms of internal organization, and that would be the cytoskeleton. So all that the cytoskeleton is internal cellular scaffolding, and that scaffolding is made of different proteins. And so you can think about the scaffolding on a building that sort of helps hold that building and shape that building and hold it up. In the same way that buildings have a scaffolding, cells have a scaffolding, and that is called the cytoskeleton. So just like you have a skeleton, cyto means cell, so this is the skeleton of the cell that kind of makes the shape. And what we're looking at here are things like these microfilaments and intermediate filaments that you're looking at here, and all these sort of yellow things that you're looking at. And that is the cytoskeleton. And we look at prokaryotic cells. So believe it or not, prokaryotic cells do actually have a cytoskeleton. And that helps determine the shape of a prokaryotic cell. But that cytoskeleton in prokaryotic cells is not dynamic. So remember, dynamic means it can move around. So the opposite of dynamic would be static, or sit still, doesn't move. So in prokaryotic cells, they have a static or non-moving cytoskeleton that's made up of different proteins that are different than those that make up the cytoskeleton in eukaryotes. And because of this, prokaryotic cells tend to be really stiff and rigid. Now, on the other hand, eukaryotic cells are very unique in that they have a very dynamic cytoskeleton that can be remodeled and moved around. And because it can be remodeled and moved around, that enables the whole cell of eukaryotic cells to move around relative to the environment. They can change shape, and they can even move items within the cell too, okay? So this allows for a lot of things to just move around and eukaryotic cells in general to be dynamic. And the sort of classic example of this is an amoeba. So just to sort of show you an idea here, here's an amoeba cell. And this amoeba cell, basically you can see it growing and expanding. So this is a single-celled eukaryotic organism with a very dynamic cytoskeleton that can sort of move and adjust as the cell moves and adjusts and moves around. And so this is something that's unique to the eukaryotes. A prokaryotic cell could not adjust its shape and size like a eukaryotic cell can because it doesn't have that dynamic endomembrane system. So let's talk about the other aspect of this internal organization that's unique to eukaryotes, and that is the evolution of a dynamic endomembrane system. So remember, dynamic means you can move around, endo means within, membranes are membranes, and so basically there's this whole system of internal membranes that are interconnected throughout eukaryotic cells. And these membranes can change sort of shape and move around all through the cell. So if you think about, for example, the nucleus is surrounded by this endoplasmic, what's called an endoplasmic reticulum. This is a series of membranes, and these membranes, or the ER, produces lipids, or they help. This is where lipids are synthesized. This is also where proteins are synthesized. You can have little vesicles sort of bud off or pop off of the endoplasmic reticulum to bring basically these proteins and lipids into another sort of dynamic membrane system called the Golgi apparatus, where it'll modify these lipids and proteins and package them into other vesicles. And so they might be packaged into these other sort of budding vesicles that pop off and secrete wastes out, or they can pop off these things called lysosomes that basically are like the stomach of the cell. They have a bunch of digestive enzymes. You have other membranes that can pinch off, bring a little piece of food in. Those lysosomes can fuse with this little piece of food and help digest it to get nutrients into and move around the cell. And so you have these dynamic systems of membranes within the cell, or endomembranes, that are sort of popping off and moving around all over the place. And you can really think of this as sort of like the assembly line of the cell, where you have different things, nutrients coming in, proteins and lipids moving around, wastes moving out, and this whole assembly line that's made possible by this organization of dynamic endomembranes. And so the huge advantage of this endomembrane system combined with a cytoskeleton is you can have very organized contents, and you can move molecules, whether it's food, whether it's certain molecular signals, whether it's proteins, whatever it is, more quickly through the interior than you could by diffusion alone. And so this allows cells to get much larger. It allows eukaryotic cells to be larger, to move stuff around actively instead of just simple diffusion and random movement of these different sort of proteins and important molecules through this eukaryotic cell. In addition, it allows eukaryotic cells to change shape, and that's really important if they need to create different cell types, which is a big part of multicellularity and unique traits that are, traits that are unique to the eukaryotes. And so, for example, you can think of stem cells. Stem cells are unique eukaryotic cells, and they're really the shapeshifters of the cellular world. So I have this picture of Mystique, the X-Men, that's a shapeshifter. She can morph into all these different shapes and make herself look like any person or anything that she wants to. Stem cells are like the Mystique of the cellular world, where they can replicate themselves, or if they want, they can shapeshift into all these different types of cells, whether it's a red blood cell, whether it's a neuron, whether it's a muscle cell, whether it's a smooth muscle cell. All of these types of remodeling that has to go into this type of shapeshifting, so to speak, is enabled by this dynamic cytoskeleton, as well as this dynamic endomembrane system. So very important innovation unique to the eukaryotes. Another important thing that dynamic membranes enable is phagocytosis. So phago means to eat, cyto means cell, and cytosis means to break apart. So this is basically cell eating is what phagocytosis means. And having that dynamic endomembrane system as well as a dynamic cytoskeleton allows cells to change shape to engulf other organisms and then combine those and break them down and move different parts of the nutrients once that organism that's consumed or that cell that's consumed is broken down within the cell. So you can see here a diagram of one organism that's sort of outlined in white and another unicellular organism that's in red here. And this is that red unicellular organism being phagocytized by this white outlined unicellular organism. And so it's being brought into this cell. It's getting surrounded by another by a membrane. So this is an example of a sort of dynamic endomembrane system bringing this in, and this will be further broken down and digested and eaten. So this helps eukaryotic cells deal with that lower surface area to volume ratio associated with larger cells by moving nutrients and wastes all around within the cell. Okay, so let's talk about some other important innovations that evolved in the eukaryotes. Once again, the Bill Gates of the cellular world. So we're going to talk about organelles, we're going to talk about a nucleus and linear DNA, we're also going to talk about sexual reproduction and multicellularity. So, organelles. What are some examples of organelles? Spend 10 seconds just rattle off a couple names of organelles for me. So lots of different examples out there. Some pretty classic examples are things like mitochondria that make ATP via aerobic cellular respiration, or chloroplasts that make sugars via oxygenic photosynthesis. So these are two examples of important organelles that are the powerhouses of different cells, so to speak. So mitochondria and chloroplasts, important subcellular organelles. And just as a reminder, answer these two questions. True or false? Eukaryotic cells have organelles, and true or false? Prokaryotic cells have organelles. Eukaryotic cells have organelles. That is true. Organelles are traits these are unique to eukaryotes. Only eukaryotes have organelles. Prokaryotic cells do not have organelles. So this is a trait that's uniquely evolved in the eukaryotes, an innovative trait that's unique to them. And what is the adaptive advantage to organelles? So why have organelles? What does that do for eukaryotic organisms? So a couple important things that organelles do. So just to sort of reiterate here, organelles, we use the example of mitochondria and chloroplasts, but there are lots of other organelles that are out there. Those are not the only organelles. And so all of these organelles, whether it's mitochondria, whether it's chloroplasts, whether it's some of the other organelles that exist in eukaryotic cells, they have these three major important advantages that they confer to eukaryotic cells. One is that chemical reactions can occur on the membranes that make up organelles. So they provide a membrane or a surface area for chemical reactions to occur instead of having those reactions just occur floating around in the sort of liquid that fills up a cell. Number two, membrane-bound organelles can maintain high concentrations of the molecules needed for specific reactions. So instead of those molecules just sort of floating around and moving around randomly all throughout or within a cell, those chemicals can be highly concentrated in one specific area so that you can have a more efficient reaction occur. And lastly, incompatible chemical reactions or chemicals can be separated into different organelles. So you can talk about reactions happening in independent places, or you can talk about individual chemicals or reagents occurring in different places. So let me just drive this home a little bit more, talking about the importance of the membranes of organelles. So chemical reactions are situated on the membranes of many organelles. And mitochondria and chloroplasts, once again, these are not the only organelles, but these are a great example of that. So if you look at mitochondria, where aerobic cellular respiration occurs, or if you look at chloroplasts, where photosynthesis occurs, and you look at all the sort of membranes that are going on the inside of mitochondria and all these sort of membranes that are going on the inside of a chloroplast, if you were to zoom in on those membranes, you'd see these yellow layers that we're looking at here and here. Those yellow layers represent the membranes, and they have all of these proteins that are embedded in those membranes. And each of these proteins plays a really important role in making these important cellular reactions happen. So whether it's the production of ATP that we have occurring here in the membrane of the mitochondria, or it's the production of sugars or energy via oxygenic photosynthesis. Once again, we have all of these important proteins embedded in the membranes of these chloroplasts. And so the more of these membranes that you have, and the higher sort of surface area to volume ratio that you have of these membranes, the more of these proteins you can pack in, the more of these reactions you can have occurring, and the more sugars you can generate, or the more ATP you can generate. And so here, once again, we've got a great example of structure equaling function. So the high concentration of membranes in mitochondria and chloroplasts relates to their function of energy production and needing a lot of membrane space for these special proteins that are involved in the chemical reactions that occur in cellular respiration and photosynthesis. Let's look at that other important characteristic of organelles, which is this idea of compartmentalization. So let's pretend you have a molecule this and molecule blue hexagon, and you have this green cylinder molecule or chemical or whatever you want to call it. So this and this react together, but this interferes with the reaction. And so if you have a prokaryotic cell where there's no organelles, there's no compartmentalization, basically you have all of these chemicals all floating around within the cell, and this green molecule or this green chemical can interfere with the interaction of these other two chemicals that are important to have together to react for some important reaction within the cell. Whereas in eukaryotic cells, you can have an organelle, and that organelle can basically specialize to concentrate and separate these chemicals. So you can put the two chemicals that are important to react together and try to concentrate them in that area, and the cell can have different apparatuses to make sure that those chemicals get concentrated within those organelles. And it can also separate those chemicals that interfere with that important reaction so that it doesn't interfere. So organelles basically prevent unwanted interactions by confining molecules to separate organelles or a separate space within an organelle, and also increase the chance that two molecules in a reaction will interact since they concentrate those reactants within the cell. Okay, answer this question. If you said E, none of the above are correct, you are right. So incompatible chemicals are combined into the same cellular compartment. This is false. It's compatible chemicals typically combined in the same compartment. There is less internal membrane to carry out essential membrane-bound chemical reactions. This is false. Typically, there's a greater amount of internal membrane to carry out. So remember, in mitochondria, there's a large concentration, lots of surface area for those membranes, so there's a lot more internal membrane to carry out those essential membrane-bound reactions. And do they decrease the chances that two molecules in essential reaction will interact by diluting them? No, they do the opposite of that. They actually increase the chances that two molecules in an essential reaction will interact by concentrating them. So none of those answers are correct. Organelles do the exact opposite of that. All right, let's look at this next piece, the nucleus and linear DNA, or genomic organization, and this important innovation that's unique to the eukaryotes and contributes to their success. So when you look at prokaryotic organisms and you look at their chromosomes, they typically have smaller circular chromosomes. So this is a very simplified diagram. This probably looks more like a plasmid. Typically, a chromosome within a prokaryote will be all sort of jumbled up, but if you're to pull it apart, make it big enough, eventually it's going to make a big hula hoop like this. Now, one problem with this is bacterial chromosomes, because they're circular, a big part of that is that they only have what's called one origin of replication. So what that means is there's only one spot within that entire chromosome where it can begin to make a copy of itself. And so you need to, if you're going to replicate a copy of this chromosome, you need to replicate at that origin, and you can only replicate from that one spot. And so because it initiates replication at a single location, replication rates are limited by chromosome size. So if you have a bigger and bigger and bigger and bigger chromosome, it's going to take longer and longer and longer and longer to replicate that chromosome, because you can only start replicating at one single place And what that means is there's actually selection for smaller genomes typically within bacteria. So a smaller genome can basically be copied much more quickly, even though there's only this single location. And that means that that genome can make more copies of it quickly, that bacteria can replicate more quickly. And if there's any extra DNA that sort of builds up in this chromosome over time, it's going to be purged, it's going to be selected against. And so selection should favor smaller genomes. Now in the eukaryotes, we have multiple linear chromosomes. So remember we learned that DNA in eukaryotes is organized into these linear chromosomes. And as it turns out, DNA replication can be initiated in multiple locations, not just multiple locations on an individual or across chromosomes, but multiple locations also within an individual chromosome. And so in this case, replication rates are not limited by chromosome size. And because of that, there's not as strong of selection against having these larger genomes. And there's also not as strong a selection against genes that are not absolutely vital for the survival and replication of these eukaryotic cells or eukaryotic organisms. So in other words, you can build up some junk DNA in there, and because DNA can be replicated in multiple locations, there's not strong selection against having that extra sort of DNA building up within the genome. So just to sort of visualize this and how this sort of affects eukaryotes, because you have multiple linear DNA chromosomes that have multiple starting points for replication, and you can have not strong selection against the buildup of extra DNA, you can actually get some buildup of extra DNA in the genome. And some of that extra DNA can become involved, or some was evolved that became involved in something called gene regulation. And all that gene regulation is the controlling of the expression of gene products. So you can think of gene regulation as sort of a light switch or a volume knob. So essentially there are these genes that can turn on and off other genes or regulate how much a gene is expressed. So when we're talking about a gene being expressed, that means, remember, genes, that's DNA, that codes for RNA, that RNA codes for proteins, and the amount, and that protein ultimately results in a trait. And so a gene being expressed means how much does that gene produce that protein? How much is that protein expressed? How much is that trait expressed? And so these regulatory genes can act like light switches or volume knobs that turn on how much of that protein is produced by that gene and how much of a trait is expressed, or if that trait is expressed at all. And that in turn, so the ability of these eukaryotic organisms to regulate their genes made way for the evolution of complex life cycles and multicellularity. So basically, allowing eukaryotes to turn on and off or up and down different genes allows them to potentially make different cell types using the same DNA code, using the same chromosome. So if you have all of the genetic information to make all different types of cells, whether it's a neuron, whether it's a muscle cell, whether it's a skin cell, if all of that information is in here, you can turn on and off particular genes to decide what a cell is going to become. But you need gene regulation to be able to do that. So just looking at this another way, gene regulation controls what a stem cell, or really any cell becomes. So you can think of all of these different chromosomes that make up your entire genome. All of these chromosomes exist in every single cell in your body. And which of those chromosomes gets turned on can determine what a cell does, what a cell looks like. So if we think about, for example, just a stem cell has all of these different chromosomes, and we can decide which chromosomes we want to turn on and turn off. So this is gene regulation, and that will express different proteins, and those proteins will determine what this stem cell, remember this is the shapeshifter cell that can become any type of cell, ultimately becomes. So that can determine whether this cell becomes a neuron, whether it becomes a red blood cell, whether it becomes a muscle cell, all of those sorts of cell types that also have a nucleus and also have all of these genes that make up our genome. So this ability to gene regulate is really important for making all these different cell types, and making different types of cells is really important for eukaryotic organisms to be able to evolve all these different types of life cycles. So this is just one example of a life cycle where you have an adult frog that lays eggs. Those eggs are obviously made up of slightly different cell types that hatch into tadpoles that are made up of slightly different cell types that turn into a frog that goes through, these go through metamorphosis, and once again, slightly different cell types. And all of this stuff is regulated through this sort of gene regulation and differential expression of proteins. And same deal with multicellularity and the ability to have all these different cell types that form tissues and organs. And so having all these differentiated cell types, you need to have gene regulation and differential expression of genes to be able to form these different cell types, which form also ultimately into tissues, ultimately into organs, and ultimately make up complex multicellular organisms like humans. So that's that idea of genomic organization. Lastly, let's talk about the importance of sexual reproduction, specifically its importance for genetic recombination. So sexual reproduction, once again, this is a unique innovation, a unique trait that's evolved in eukaryotes, and only eukaryotes can reproduce sexually. Remember, we talked about prokaryotes and horizontal gene transfer for generating genetic variation, but with eukaryotes, they use sexual reproduction. And once again, that's a trait that is unique to the eukaryotes. So in sexual reproduction, basically you have sex, where you have haploid or 1N cells fusing to make diploid or 2N cells. You can have different organisms that do this in lots and lots of different ways. Sex isn't just the way that we see it in humans, where you have a mom and a dad making an egg and a sperm, but we'll talk about human reproduction. And the important thing here is that sexual reproduction, when you have sperm from a dad and an egg from a mom, basically you're recombining the different alleles that exist into unique combinations in the offspring. And this is why you're not exactly like either parent. So your dad has his unique set of chromosomes and alleles associated with those chromosomes, and mom has all of the same chromosomes, but she has her own unique alleles on each of those chromosomes. And so when you combine them, you can get different combinations of alleles to produce the offspring. Now, on top of those sort of unique combinations from mom and dad, meiosis also plays a really, really important role. So meiosis basically takes diploid parents and cuts their diploid number of chromosomes in half. So you go from 2N to 1N to get these haploid gametes. And those gametes can have lots of different combinations. So you don't only just get the combinations that dad had, but you can get unique combinations in the gametes that even the dad didn't have, in the sense that the alleles that are on the chromosomes can combine in new, unique ways through what's called independent assortment. So I don't want to get into all the details of this, just suffice it to say, meiosis makes even more unique haploid gametes, and those haploid gametes can combine in unique ways to make, even more unique or different offspring with every fertilization. So this is why you're not exactly like your siblings, because the combination of genes that you get from your mom and dad are not going to be the same every single time. You'll get unique combinations thanks to this process of meiosis. And so what's the adaptive advantage to reproducing sexually? Why is this important for eukaryotes? So number one, we pointed out that sex combines different alleles from different parents. So this is overall going to increase the genetic variation in the second generation or in the offspring. You're going to have unique combinations of alleles, so more genetic variation in the population. And in addition, meiosis puts a second layer. Meiosis is a process that, once again, it's unique to eukaryotes. It's unique to sexual reproduction. And that creates haploid cells with even more genetically unique allele combinations through crossing over and through independent assortment. And so I don't want to get into all of the details of how that works, just suffice it to say that crossing over can make these new sorts of combinations within the chromosomes, where you have them exchanging information, resulting in new allele combinations. And you also have this thing called independent assortment, where essentially the gametes that you can create can vary just depending on how things sort during meiosis. So here you get the blue alleles together, the blue and the red alleles together. Here in this combination, we've got blue and red together and red and blue together. And this is just by chance. And so that once again creates new combinations of alleles, which increases the overall genetic variation in the population. And so these two processes, sex or fertilization and meiosis, create more genetic variation. So you have offspring with lots of unique allele combinations. And the important thing here is that the alleles themselves are not unique. Those are alleles that already existed in the population. It's that you're getting new combinations of those alleles that potentially did not exist before. So you're combining those alleles in new ways. And ultimately, that increases the overall genetic variation and phenotypic variation in the population for natural selection to act upon. And so this simplified diagram sort of demonstrates that, but it really doesn't demonstrate what's going on with meiosis. But you can see here, for example, in this first generation, we have these different combinations of alleles. You can think of the green allele and the yellow allele. And now we have individuals in the next generation, we have some individuals that have only the yellow allele. So now we have a new combination of alleles that exists in the population that wasn't there before. So, which of the following is true? If you said C, you are correct. So A is wrong because the diversity of shapes is thanks to the dynamic cytoskeleton and the ability of the cytoskeleton to move around. When we're talking about the ability of eukaryotic cells to move, engulf other cells, and change shape, we're talking about the dynamic cytoskeleton and the endomembrane system and that system of internal organization that's unique to the eukaryotes. D is false because genetic diversity is generated by sexual reproduction in eukaryotes. Although eukaryotes can do or reproduce asexually, asexual reproduction does not produce a lot of genetic diversity. It basically creates genetic clones. The cell wall helps enable eukaryotes to support their larger cell volume. No. Organelles, a dynamic cytoskeleton, and an endomembrane system that are sort of moving nutrients and waste, moving nutrients around within the cell so they can get to all the parts of the cell they need to, and moving waste out of the cell so they can diffuse out before they become too toxic. Help these larger cells with a lower surface area to volume ratio exchange nutrients and waste with the environment.